We have studied the structure of muscle, which is actually made up of muscle bundles, fascicles, and muscle fibers, myofibrils, and sarcomeres. And the sarcomeres are actually units that are made up of different types of filaments, actins, myosins, tropomyosins, and troponins. And now we are going to study actually the mechanism of muscle contraction. And the mechanism of muscle contraction takes place in a different events or steps. There are certain requirements for muscle contraction. For example, it requires nerve impulse. For example, it requires calcium ions. It requires energy. And finally, the twisting of filaments and overlapping and sliding of filaments to constitute the contraction of muscle fiber. How this each event takes place and what are the actual details of every step, we are going to study them one by one. The very first step in the contraction, the mechanism of muscle contraction is the requirement of nerve impulse. And nerve impulse leads to a muscle fiber with the help of motor neurons, motor endings. And how actually this is conducted, how impulse is conducted from neuron to a muscle, it actually is in a structure that we are going to study today. And this is how we would say impulse travels from terminals of the neurons to the sarcolemma of a muscle fiber. And it actually is taking place at a place to which we also call it's actually a neuromuscular junction. It's going to be a junction where neurons are going to make a junction with a sarcolemma to conduct impulse inside sarcolemma. What actually is the structure and what are its parts of this junction? The first part of this junction is axon terminals. This is axon and this is axon terminal. And this axon terminal actually is having a membrane. That membrane is called neurolemma. And neurolemma is a membrane of neuron, and it is polarized, as we know. It is positively charged externally and it's negatively charged internally. It is its resting membrane potential. And when it comes into action, there are certain channels such as sodium and potassium and pump, sodium potassium pump, etc., that actually provide ability to this membrane to change its potentials, especially when a stimuli strike. Membrane is depolarized, it is repolarized, and it regains its potential again and maintains resting membrane potential. Actually, this neurolemma contains variety of gates here. They can be sodium gates, they can be potassium gates, they can be sodium potassium pump. Actually, these gates help it to get depolarized, and that depolarization actually is a nerve impulse which is to get conducted from terminals to a sarcolemma. And this terminal has a special kind of secretory neurotransmitters. To these neurotransmitters, we say these are said to be acetylcholine, which is synthesized actually in a neurons as a kind of a neurotransmitter is called acetylcholine, that is in form of vesicles present here, and this acetylcholine would diffuse and would come in a cleft, and that would finally be received on receptors on a sarcolemma, and that will induce depolarization right in sarcolemma. So these are vesicles, these contain neurotransmitter, and this neurotransmitter is called acetylcholine, where acetylcholine comes from mitochondria, which then combines with the acetate and it synthesizes acetylcholine, and that acetylcholine in forms of vesicles finally diffuses to the neurolemma and it leads to a cleft and goes onward. What actually happens, how depolarization is transported or conducted from this terminal regions to a sarcolemma, there are certain steps. Before starting that steps study, this is structure of muscle fiber. This muscle fiber is having external membrane. This membrane is called sarcolemma. Inside sarcolemma, this is called sarcoplasm. And within sarcoplasm, there would be a reticulum called sarcoplasmic reticulum. And these are myofibrils. And each myofibril is actually made up of muscle fibers. Actually, each myofibril is made up of sarcomeres, which is made up of different kinds of filaments. So this actually is a structure of muscle fiber. And the structure of muscle fiber, where it's actually going to have a junction with the terminals, it forms a plate that is called end plate. to this region, we would consider skull end plate, where the sarcolemma is gonna be depolarized. So this is an end plate, and on this end plate there are different kind of receptors present. These are called different kinds of receptors. Actually, these receptors are gates also, and they would open up and they would help the movement of sodiums from outside to inside, and they would depolarize to this membrane. But actually, these gates are not sensitive to voltage like the gates of a neurolemma. Actually, these gates are chemical sensitive gates. They are called ligated gates. They are ligated gates, and they can be sensitive to a kind of chemical such as acetylcholine, such as nicotines and etc. And when these neurotransmitters get received to these receptors, they actually open up these gates and they flow the sodiums in, and they would cause the depolarization in a sarcolemma that would conduct throughout muscle fiber. So these are the structures, these are called receptors that are present on the sarcolemma, and they will open up the gates. They can be called ligated gates, there can be chemical sensitive gates, they will constitute the depolarization here. This is a space between these two, and that is called cleft. Now we are going to study what actually happens, how this impulse is conducted from terminals inside sarcolemma or muscle fiber. As depolarization reaches to a terminal, what actually is depolarization? Depolarization is actually change in the potential of membrane, which externally is positive, which internally is negative. As depolarization reaches to a terminal, depolarization actually is opening up of the sodium gates. These voltage gates open, and these voltage gates are sodium gates. And when these sodium gates open, sodium starts moving from outside to inside. And as sodium gets open and sodium start moving inside, from inside where membrane is negatively charged, that membrane would start becoming less negative, and it will become zero, and then it will become positive. And when membrane is internally positive, membrane's poles are changed and membrane is depolarized. And to this stage of neurolemma, we would say neurolemma is now depolarized as it is internally positively charged, which previously was a negatively charged when it was in a resting state and it was having its resting membrane potential. But as stimulus reaches here, impulse reaches here, it depolarizes the membrane, and as membrane is depolarized, it actually opens up these other channels and proteins which carry calcium inside terminals. From cleft, calcium moves inside these terminals, and calcium actually binds to the specific receptors that are present on the membrane of the vesicles. And now the receptors that are present also on the vesicles, on the membrane of the vesicle, and the same receptors are also present on the neurolemma. They actually help these vesicles to move and to get diffused with the neurolemma. So what actually happens? Depolarization reaches its place. It gives permeability to calcium carriers, and they carry calcium inside terminals. And when calcium moves inside, it actually makes the vesicles of a neurotransmitter move, and they get diffused with the neurolemma. And as they diffuse with the neurolemma, they release acetylcholine inside cleft. And as acetylcholine comes in a cleft, it now has to get attached with a specific kind of receptors that are present on sarcolemma. Remember, initially sarcolemma is externally positive and it's internally negative, as it's maintaining its resting membrane potential, or it's in a state of rest. Or as acetylcholine reaches to these receptors, and these are sensitive to these receptors, acetylcholine will remain attached to these receptors, and as it attaches to our receptors, it actually opens up these gates. And when these gates are open, these are called ligated gates are open, sodium starts moving from outside to inside. And as sodium starts coming inside, the internally membrane, which initially is negative, as sodium will start coming inside, the internal membrane would start becoming positive. And as membrane will become positive, membrane actually, which we say sarcolemma, is depolarized. And this depolarization actually is a conduction of impulse that is conducted now in a sarcolemma. And this depolarization will now travel onward, and in order to get spread throughout muscle fiber, it will also reach to a transverse tubules, and transverse tubules would carry to this depolarization to hold this muscle fiber. And as this nerve impulse or depolarization is reaching to the whole muscle fiber, it will start leading to the other events that are required for the muscle contractions. And one such important event is the release of calcium ions. See, as this sarcolemma is depolarized, this depolarization results in the release of calcium ions. Depolarization stimulates to a sarcoplasmic reticulum, and sarcoplasmic reticulum releases calcium ions. And when these calcium are released, these calciums move from sarcoplasm inside myofibrils and they move inside sarcomeres. And as calcium reaches to a sarcomere, it is going to lead to the other important functions of the muscle contractions, which we are going to study later. As calcium is released, it will attach to a troponin, and it has to twist the position of tropomyosin, which would actually open up expose the sites of actin where myosins will get attached and contraction will begin. But that is another event that we are going to study in another video. So wait for the next video, where we are going to study what actually happens to a calcium, where it binds, and how filaments actually twist, and how actually muscle filaments slide over each other, and what actually contraction is. We are going to study it in a next video.